Hello, everybody. I think it's time now, so the others will continue joining, at least maybe during my introduction. So, am I audible for everyone, please?
Yes, you are.
Thank you so much. Okay, so good afternoon, everyone. And maybe we can say good morning for those who are joining from the west part of the world. First, I want to send you the apology of Dr. Abdullah Yassin, who was supposed to be seen here today as a moderator. I wish I can represent him as it should be. I am Abdullah Muhammad Ibrahim from Abrar University. So this is our third meeting about our webinar series on prioritized zoonotic diseases in Somalia for the year 2023. This topic, of course, very special for Africa and particularly for East Africa. So today we are going to hear about anthrax. And before I complete the title officially, maybe anthrax have many names according to different geographical areas, endemic areas. For example, at least in Somalia, I hear they call it Kud, and Arabic people, they call it Al-Jamra al-Khabitha. So it is a very important disease. So the topic today is anthrax at the human-animal environment interfaces. And allow me to say that who is better than our speaker today, Dr. Siobhan, at least in East Africa, to speak about this disease? Maybe you will agree with me when I tell you briefly about our speaker today. Dr. Siobhan is Associate Professor of One Health at University of Liverpool, United Kingdom, jointly also appointed at International Livestock Research Institute in Ethiopia. Prior to this, she held several academic appointments in United States and Australia. Dr. Siobhan has double background, both veterinary medicine background and also she added public health background, and those are the people of One Health always doing that. She has been based full-time in Ethiopia since 2018, and during this period, she has served as research leader for Horn Project in Ethiopia. She also served as ILRI principal investigator on HEAL project, and also Ethiopia country coordinator for COHESA project. And as you can see, all these projects are just house of One Health and zoonotic diseases issues. Therefore, I said, who is the best to talk to us today about anthrax? So thank you again, everyone, for joining us. And I want all the participants enjoy what the professor or the doctor is going to tell us about the disease. And all of you, thank you. And so Professor or Dr. Siobhan, the floor is all yours. Thank you.
Thank you very much. Let me just move my screen. Okay, I trust you can see everything correctly, yes?
Yes, it's appearing very clear.
And is my audio okay? There's a bit of background noise from my side.
Yeah, everything is okay.
Okay, great. Well, thank you very much for joining on this Wednesday afternoon. I'm sitting in Ethiopia. I'm just going to mute you. Yeah, I think that's better. There's some feedback. As you just heard, I'm sitting in Ethiopia and have been here for about the last 5 years. However, I am Australian by background, and anthrax is a disease that we experience in Australia as well, and certainly an important disease. I have to let you know that I am by no means a specialist when it comes to anthrax, although it is a disease that has always fascinated me precisely because it has such strong connections to human health, animal health, and environmental health, and really so sensitive to the environmental aspects of the disease. And so therefore, I think it's a great disease to learn more about and to go into if you're particularly interested in One Health, because you're naturally working across all three of these human, animal, and environment interfaces. So I have been told that I should speak for around 45 minutes. And so what I have prepared for you is really an overview of anthrax more broadly, thinking at the global level. So by the end of the presentation, you should have a clear understanding of what causes anthrax, how it's transmitted, the types of clinical disease that it causes, diagnosis and treatment, and prevention and control. And of course, I'll be talking about the human and the animal aspects there. I'll then talk briefly about the epidemiology of anthrax in Africa, but I'll tell that through a case study that I was involved in writing up from Ethiopia, which really speaks to the nature of these outbreaks that are experienced in Africa, and I suspect it would be very similar in Somalia. And then I'll finish by sharing what I've been able to find about what is known about anthrax in Somalia. Of course, I'm sure there are people on the call who've seen cases in Somalia, who've managed cases in Somalia. So again, I'm by no means an expert, and I hope by the end of the session, we'll be able to talk about your experiences as well. But what is known about anthrax is really about what's been published about anthrax in Somalia. So just moving straight into an overview of anthrax. I think it's important to realize that it's a disease that's principally a disease of herbivores, but it is a disease that affects all warm-blooded animals, and that's particularly true of herbivores. And humans, of course, being warm-blooded animals, are susceptible to anthrax also. So to give a quick summary of what an outbreak typically looks like in an African kind of context, there might be a sporadic outbreak, typically manifested by sudden death in herbivorous animals, and they may be livestock, or they may be wild ungulates, so hippopotamus or antelope. And there may be a few cases in carnivores. It's not very common, but that tends to happen because these carnivores will predate on the herbivore carcasses. And then there might be a few to many cases of humans who contract the illness through contact with animals or their animal products associated with these particular outbreaks. So that's typically the sequence of events. There would usually be an outbreak in animals, and that would then be followed potentially by cases in humans, depending on how well an outbreak is managed and the types of human behaviors that happen in between those two things. So anthrax is a bacterial disease, and it's caused by a bacteria called Bacillus anthracis, which is a gram-positive rod. It's sort of the quintessential gram-positive rod. When you're learning microbiology, this is what you'll learn as being the example of a gram-positive rod. And it's naturally occurring in the soil. So its natural state is being spores that lie dormant in the soil. And it's only once they are consumed or somehow enter the host that they become active, they multiply, and then cause toxins. And it's these toxins that cause the disease in humans and animals. As I've said, all humans and animals are susceptible, but particularly herbivores. So as I said, I come from Australia, which is a country that experiences sporadic outbreaks of anthrax. Largely, those are limited to livestock. It's very rare to see anthrax cases in humans in Australia. But there are other parts of the world that ecologically are very vulnerable to, or rather suitable to anthrax as well, with China, Kazakhstan, North America being the other global hotspots that are recognized as being important. This map was produced using ecological niche modeling. So that uses a range of different inputs to try to predict the suitability of habitat and where the disease might be happening. But of course, that model is informed by quite limited data from Africa. You can see that there's some hotspots down in Southern Africa and West Africa, and the Horn of Africa region and East Africa would not appear to be hotspots according to this suitability model. But that's in fact not really the case. We do know, of course, that it is a disease that widely occurs throughout much of Africa. And so this is another map, not based on modeling, but based on reported cases and perceptions by WHO on the endemicity of anthrax. So you can see in Africa, neighboring Ethiopia, where I'm sitting, would be considered a hyper endemic area, so an area that experiences epidemics. Again, Southern Africa, so Zambia, Zimbabwe, Botswana. A lot of research on anthrax in Africa comes out of those three countries. Also Chad and Western Africa, countries in Western Africa are recognized as being hotspots amongst the African continent. And it is seen and regarded as a top priority zoonosis across much of Africa. So where countries have done these multi-stakeholder meetings and come up with the top priority diseases, anthrax is often one or two, but certainly in the top 5 for most countries. And that does include Somalia. So I'm going to dive straight in and talk about the environment up front, which is what we tend not to do with One Health, right? We often talk about humans and then animals and then environment. But for anthrax, the connection to the environment is just so strong that it's important that you understand this cycle that really dramatically involves the environment. So the spores, as I mentioned, is the natural state of the organism, and they are highly resistant to environmental degradation. And so they can survive in soil, depending on the conditions, for months to years to even decades. And I'll give an example of that in a second. And all of these things are affected by the temperature, humidity, and the soil conditions. So that's how they can use the ecologic niche modeling and use a variety of different environmental conditions to model the suitability of anthrax across the planet. And unfortunately, it's not as simple as saying anthrax happens following wet conditions or it happens following dry conditions, because there's so many different factors that influence its persistence and its emergence from the soil. So it's associated with wet conditions, largely because moisture in the soil will be favorable for spore germination, but also flooding can essentially contribute to the distribution of spores throughout the soil and can contaminate water sources. So that in itself can contribute to outbreaks. And so in Ethiopia and Southern Africa, I believe it's principally wet conditions that are associated with outbreaks. But again, that's not consistent throughout the world. And that's because dry conditions can also change behaviors. Right, that means that particularly in pastoralist contexts, but even in other contexts, animals will be congregating around water points that are more scarce, and then that means that there'll be high densities of livestock. If one of those livestock die from anthrax, then that site will become contaminated, and more animals will be exposed. So dry conditions themselves, just by the nature of changing animal and potentially human behavior as well, can also precipitate in outbreaks. Now, I mentioned the fact that it can survive in the soil for months, years, or decades. And one of the most interesting outbreaks that I've seen reported relates to the Arctic Circle. So this is in Siberia, and they have experienced outbreaks after decades of years, after having never seen outbreaks for so many years. And that's because the frost, the permafrost, is melting, and some of the traditional burial grounds that were where they would bury reindeers that had died, they are defrosting, and the spores are germinating, and then the reindeer are accessing the contaminated soil again. So this is brought to our attention that climate-related changes are really likely to have quite a significant impact on the distribution of anthrax and the vulnerability of areas to anthrax. This was something that we modelled a few years ago. On the right side, you can see this is actually the planet looking down, so the northern hemisphere, with the US being over this side. If you can see my pointer, I don't know if you can see my pointer, can you? You can? Yes. So this is the US, this is Canada, for instance, here's Europe, so you're looking down. And you can see currently, although those hotspots are well, showing up in the figure in the top, when we fast forward to climate predictions into 2050, you can see that much more of the US is now vulnerable to anthrax outbreaks. The suitability has increased a lot more because of this changing climate in those areas. And this is, of course, warm and arid areas. So it's not only these frosty areas that are going to, like in the Arctic, that are going to be vulnerable to outbreaks. So once again, just to reinforce the really strong connection to climate and soil conditions. So let's now think about how that translates into transmission in animals. So in the animal context, animals become infected by ingesting spores from either the soil or the water or potentially plants. They can also acquire the infection through inhaling spores. If you can imagine a herd of herbivores running through savanna, there's a lot of dust that gets aerosolated, and if they run across an area that contains anthrax in the soil, there's going to be potential aerosolization of spores. And so when animals or herbivores take in the spores, you'll see soon, it will cause rapid death. And it's this death that then leads to inoculation of the soil. The spores exit the body through bleeding from the nose in this case, but from all orifices. And then the spores re-enter the soil. And there's some really interesting research in recent years which have really spoken to the interaction between plants and soil conditions that really promote the stability and resistance of the spores, but also it can contribute to plant growth. So in areas where there is anthrax, the plants themselves have been found to be quite different. And those plants, in turn, support the optimal habitat for the spores. So again, that really strong connection between the environment and the animals in this case. So once animals become infected, the main way that they will experience illness is through sudden death. And I apologize, there's not going to be, most of the photos in this presentation are not going to be very nice. You can see here cows that have died from anthrax, and the characteristic finding would be that they have blood oozing from the body. And this is again important for the cycle to continue. The spores exit the body and go back into the soil, and it's when another animal comes along, grazes in that area, that they will become infected. One of the other characteristic findings is that animals tend not to develop full-blown rigor mortis. So on discovery, they are often bloated and they won't have developed rigor mortis, and they will often decompose very quickly. Other findings would be things like swelling and edema that would be visible externally. And also, if you were to open up the carcass, you would find internal bleeding. But as I will stress shortly, it's really recommended that you not open up any carcass that has been suspected to have died from anthrax. I mentioned there sudden death. Sometimes there might be animals that experience clinical illness before death, and oftentimes that will be respiratory distress. But usually animals will simply just appear dead, and owners will find them, and potentially that would be reported to authorities. So how is it diagnosed? It's really important to understand the dynamics and the history of anthrax, because that's going to be important to clue you in that an animal might be potentially infected with anthrax. So typically, as I say, animals would be, it's a sudden death kind of scenario. And so you'd be developing a list of differentials based on that. But if you know that this is happening in an area with a history of anthrax outbreaks, then that's going to be important. In animals, it's recommended that you collect blood from peripheral vessels and make an air-dried blood smear. But at no stage should you open up the carcass and conduct a normal, a post-mortem, because in doing that, you're going to expose the environment to the spores, and that will create greater contamination. And that's to say nothing about your own personal risk as a veterinarian who's doing that. There are various types of laboratory tests that are available in animals. Most commonly, blood smear and microbiological culture would be used, but also PCR confirmation is often used depending on the country. So again, to reinforce, if you are approaching an animal with suspected anthrax, you need to use appropriate PPE. You can see here the veterinarian is taking a swab sample from the nose in this picture, I think, and she's gowned up properly, wearing PPE, including respiratory and gloves protection, cutaneous protection. Treatment in animals is unusual because, again, oftentimes they are found dead. But I'm surprised in Ethiopia to hear how often they will attempt to treat animals with penicillin antibiotics. So in many respects, that would be used as a post-exposure prophylaxis. So in animals that are likely to have been exposed to the animal, maybe in the same herd or flock, that might be used. But more often, vaccination will also be done. So ring vaccination around animals that are in that vicinity. One of the important things to note is that animals that are treated with antibiotics have a delayed development of immunity. So it's important to delay vaccination for at least a week after antibiotic treatment. Otherwise, they will need to be vaccinated again as a consequence of antibiotic treatment. But as I say, generally treatment would not be pursued. You wouldn't have the opportunity to pursue treatment. So prevention and control is then really critical, and early detection of the outbreaks in animals is going to be especially important to prevent further exposure to other animals, as well as environmental contamination. So prevention and control is usually enacted through vaccination. Again, in the Ethiopian context and much of sub-Saharan Africa, it will be a ring vaccination following an outbreak. But in other countries where there are adequate resources, vaccination might be used as a pre-exposure measure. So in Australia, there will be annual vaccination of livestock that reside in the anthrax belt, what we call the high-risk area within the anthrax, within Australia. And that will be a way to prevent the infection from emerging in animals in the first place. When responding to an outbreak, environmental management is really critical. And I think this is an area which is not necessarily done well in the African context. So proper disposal is very important. Typically burning would be recommended, burning on site and then burying the ashes. If that's not possible, then burying the carcass very deep, several meters down is recommended. that is generally okay. If veterinarians or authorities can manage a situation, disposal can be done properly in this context. But I think there's less practice of decontamination. So that's where something like formaldehyde would be applied to the areas and the area marked off so that future animals can't come and graze in those areas. Again, I think that's a weakness in the response of most sites in Africa. And then it's also very important to prevent scavenging of carcasses. So in this context, most oftentimes that would be placing thorn bush or some deterrent in that area. In fact, formaldehyde can also deter predators for going into that area as well. So that's another important consideration. When it's really impossible to dispose properly, another recommendation is to let the carcass decompose. So you would effectively try to keep it, keep other animals away from it, and that includes herbivores as well as predators. And then you allow the carcass to decompose fully, and then you might go back to try to bury and so forth. But of course, when dealing with an animal outbreak, public sensitization is going to be particularly important to prevent further transmission to humans. So I sound somewhat critical of the environmental management, but I also fully need to acknowledge that environmental management in practice is really difficult. And this is an example of an outbreak that affected, in this case, 92 hippos and 9 buffaloes. But you can see below that Uganda has experienced outbreaks of 300 hippos suddenly dying in the context of a land and water kind of environment. So you can imagine that trying to dispose of those carcasses in that kind of environment or in that kind of context is going to be next to impossible. So in these contexts, it's really, really hard to manage the carcass, and the public sensitization and keeping people and their animals away from the decomposing carcasses is going to be the really key intervention in that context. So ideally, the story would stop here. It would just be a disease that is managed in animals, that affects animals, and there wouldn't be any transmission to humans. But of course, that's not the case. So let's now shift gears and talk about how the disease manifests in humans and how do humans acquire the infection. So for humans, the spores are really entering in a similar way. It's through cutaneous or mucosal contact with infected animals or contaminated animal products. So now we introduce things like meat and other types of products into the picture. So cutaneous contact is the most common way that people will be exposed to anthrax. But they can also ingest spores or germinated cells through animal products, and they can also inhale spores. One of the really interesting case reports that you might see in countries in Europe, for instance, that don't necessarily experience anthrax outbreaks are amongst people who are drummers, and they have an African drum that is covered by a skin of a hide, like the hide of an animal that has been then exported into Europe. And those people develop either cutaneous or inhalation anthrax because they're drumming and the spores get aerosolized from the hides. So there's quite a number of different reports in the literature about this scenario. Of course, in this context, most people are going to be exposed through eating raw or undercooked meat or through having contact with the hides or the animals themselves. So people who are involved in processing hide or wool would be the people who are most at risk, as well as people who are butchering animals or contacting animals that have died from anthrax. Another name for anthrax is actually wool sorters disease because it was associated with people who are involved in the wool industry and who would develop sores on their hands after handling wool. The other major risk category are people who are exposed to laboratory cultures, so laboratory technicians who either knowingly handle anthrax cultures, but more often not knowingly handle anthrax cultures. And also, if you recall, after 9/11 in the US, there was a bioterrorism scare where anthrax spores were disseminated in the mail. So anthrax is actually recognized as being one of the key, or I guess top priority organisms when it comes to bioterrorism risk globally. And that's again coming back to this environmental resistance. The fact that it can persist in the environment is what makes it an ideal candidate for bioterrorism. Important to note that people with anthrax are not contagious, so they can't transmit the infection to another person. And when people are exposed, the incubation period can take some time to manifest, or it can manifest quite quickly. So it could be a day to a couple of months before people develop signs. Generally, depending on the resource that you consult, people will describe 3 to 4 forms. And in this African context, it would only really be the 3 forms that you would need to worry about: cutaneous, gastrointestinal, and inhalation. And all of these are represent the ways that people acquire the infection. So if they have skin contact, they'll develop lesions on their skin. If they ingest the spores, then they'll develop gastrointestinal signs. And if they inhale the spores, then they'll develop inhalation anthrax. This injection anthrax is one of the additional forms that have been described, particularly in Europe, largely among drug users who've used contaminated needles. It has not been reported in the African context and even in the North American context. But I guess in theory, it's possible that contaminated needles in the African context could transmit the infection. And you would expect cutaneous manifestation, but then more serious septic disease. So they would very quickly progress to septic form. I'll explain in detail the signs and symptoms for each of these characteristics in a second, but it's important to note that all of them can become serious if they're not treated. So it's really important that early detection happens. And again, most of the burden of early detection falls on the animal sector. If the animal outbreak is contained and it minimizes the exposure in humans, then there'll be early detection in humans that were exposed, and there'll be the prevention of subsequent cases in humans. So the cutaneous form is the most common form, and fortunately it has quite a low case fatality rate, which is only about 10% if untreated. And it's even lower than that if it can be successfully treated. And the top figure there shows the typical manifestation. So this sort of blister or bumps that will develop, particularly on the site of contact, and site of contact or the lymph nodes that are involved in the infection as well. And then it will develop this black eschar, as it's called, this black center. So it's quite pathognomonic to identify these black eschars, and quite often presumptive diagnosis can be made on the grounds of historical exposure plus the symptoms that you see there. But you can see that cutaneous forms can also manifest very severely. And here on the lower picture is a person who's been exposed with the main manifestation being around the eye. And so there's a lot of edema and swelling related to the infection of the eye as well. Other common sites would be the arms, the legs, the neck in particular, and again, the lymph nodes can become inflamed and edematous. Gastrointestinal, it's less common but more severe, and so people will typically present with flu-like symptoms, so fever and chills, maybe sore throat with swelling of the neck. On the right-hand side, you can see the manifestation of oropharyngeal anthrax. So here the infection has just been contained to the oropharynx, and there's ulceration of the tonsils and other areas in that region. But typically, people with gastrointestinal anthrax will go on to develop nausea and vomiting, and diarrhea, and that may be bloody because of internal hemorrhage. They might also develop severe pain, such as headache and stomach pain, as well as swelling of the abdomen. But again, there's sort of a dual phase to that picture. The early stages are really just flu-like symptoms, so people might not necessarily recognize or connect that they've got actually quite a serious disease, and that it might turn into something. It might just simply seem like flu. The inhalational form is the most severe form, so very high fatality rate. Around 75% of people with inhalation anthrax will die, but it's also quite rare. And as we've said, this is more the anticipated form that might happen if it's a bioterrorism incident, for instance. But generally, it doesn't occur apart from these very unusual circumstances, like a person playing drums. Again, the manifestation initially is like flu-like illness, so fever and chills with progressing to respiratory problems, so chest discomfort, cough, and shortness of breath. There might be gastrointestinal manifestations in these patients as well, and it will rapidly progress to organ failure and death in these patients. So as I've said, diagnosis is really important. Quick diagnosis is really important, and it's the clinical history that is often going to be what helps the clinician make the distinction between flu-like illness and another more important problem. The typical samples that might be collected are cutaneous samples, so skin swabs, stool in the case of someone with a gastrointestinal manifestation, and then potentially respiratory swabs or sputum in the case of people with suspect inhalation. Laboratory testing, the same as animals, so gram stain, microbiological culture, and PCR would be the main types of tests that would be performed in most contexts. And as I've said, early diagnosis is critical. So fortunately, people who do receive treatment early on will be cured. So the typical antibiotics would be ciprofloxacin and other penicillin-type antibiotics. And in some cases, antitoxin will be given, but generally that's not available, and certainly not available widely to populations, even in high-income countries, much less low-income countries. So prevention and control in humans, once again, really reliant on the management of outbreaks in animals. There is a vaccine, but it's not widely used in humans. It's typically reserved for use in high-risk populations, so military personnel, people who might be exposed to bioterrorism, and laboratory workers. But generally, the anthrax vaccination is not used. In Australia, veterinarians, for instance, would not be vaccinated against anthrax. It's not widely used at all. And so that's why raising community awareness is going to be the number one preventive in the humans, and particularly in this context, the danger of eating meat from animals that have died from anthrax. So let's move now to put this in context, and I wanted to talk through a couple of outbreaks that, as I mentioned, that I was involved in helping to write up. And these outbreaks are by no means unique to Ethiopia. If you read the literature, particularly from Southern Africa, where a lot of the outbreaks have been written, it's a very, very similar story in terms of how people were exposed and the type of scale of the outbreaks that were experienced. So this is a paper that we published, was it last year or the year before? I can't remember. But I'm happy to share with you, but it's also available online. So in outbreak one, both of these outbreaks were in Amhara region, which is in sort of central northern Ethiopia. In the first outbreak, it was a single cow that had died, and fairly uniquely, it was an animal that had been identified as having experienced respiratory distress within the 24 hours before it died. And this happened following a period of heavy rain. So if you recall, this idea of spores probably being mobilized through flooding would have led to the redistribution of spores and potential exposure of the cow in this case. Importantly, in this outbreak, it was the non-fasting season. So that means that people are eating meat in this time of year. The first case in humans was reported only 3 days later. But unfortunately, in this particular context, there was delayed reporting from the regions where this was happening, Amara region, up to the national level. And it's really the national level that has the capacity to mount a response and also conduct diagnosis using PCR. So the time span between the cow dying and actually this multi-sectoral team being deployed to the site was actually a couple of months later that the team was mobilized. When they investigated, they found that the owner of the index animal had opened up the carcass and they had dressed the meat and distributed the meat within the village. So they had either distributed it to family or sold it within the village for human consumption. And as is often the case, because people don't necessarily know any better, the hide, the bones, and the offal were just simply discarded around the environment, including in water bodies. And so you can probably imagine how this went. When they did the final assessment of the epidemiologic investigation, they discovered that 49 cows had suddenly died, and that was associated with suspected anthrax. 2 dogs and cats had died with possible anthrax. That's a bit unusual. Dogs and cats are generally considered a bit more resistant. But the fact that they were found having died after a short period of illness in the same vicinity of animals and livestock that had died was what led to this possible diagnosis. 55 people had contact with that single animal through butchering and skinning and handling of the animal. And 22 of those people went on to develop illness. And fortunately, most of those were just developing cutaneous anthrax, but 2 of them went on to also develop the ingestion form and died. And in this case, they were able to collect 13 blood samples from the 22 people with suspected anthrax, and 3 of those went on to be confirmed from the blood samples. And they also took swabs at the same time, the cutaneous lesions, but none of those proved to be PCR positive. So that was a very large outbreak involving many cows, many people, and quite a significant delay in terms of the time of the outbreak and the follow-up investigation. The second outbreak started much the same way. In this case, it was a cow and a goat that had died within days of developing some sort of non-specific illness. It also followed a period of rain in Ethiopia, but very importantly, it happened during the fasting season. So people in Ethiopia during the fasting season do not eat meat. At the time, there was, however, one suspected death related to the outbreak. So this was a person who had died. They had a cutaneous lesion that was consistent with anthrax, and they had also developed clinical signs consistent with gastrointestinal anthrax in the days leading up to their death. Once again, it was a multi-sectoral team that had mounted the response, again involving regional and federal officials, but this time a much faster time to detection and reporting and follow-up. And in this case, once again, the carcass of the cow and the goat had been shared by the community, but importantly, the meat had not been consumed. Instead, it had been dried for later consumption. And this is typical because of the fact that it was the fasting season. But once again, the hide, the bones, and the offal had been discarded around the environment, including the water bodies. In this case, there was no further animal cases, fortunately, but there were 66 people who had still been exposed through handling of the meat for later consumption. 18 of those developed only the cutaneous form, so no further gastrointestinal cases and no further deaths. But quite interestingly, swabs taken from the dried meat from the goat, in this case, did come back PCR positive, so it was a confirmed outbreak. And this is one of very few reports of detection in dried meat, actually. It has not been widely reported that that's possible. But again, what we know of the environmental persistence and stability of anthrax is consistent with that finding. So in terms of the response and how these outbreaks were handled, it was quite similar. There was recalling and burning of the remaining animal products, but clearly in the case of outbreak 1, the meat had been distributed and consumed. So the recalling of the animal products was more important in outbreak 2 because people had taken steps to preserve the meat because it was the fasting season. They then conducted ring vaccination around the areas that were affected in the livestock and launched a mass education campaign for the community. Some of the delays that were experienced, and again, this is quite typical throughout outbreaks reported in different parts of Africa. The delayed reporting to authorities meant that there was a delayed investigation. That meant that it was very difficult to obtain samples for confirmation. And it also meant that samples from particularly the cutaneous lesions were falsely negative. Clearly the people had had anthrax, but by the time of the sampling and swabbing of the cutaneous lesions, they weren't able to detect the bacterium. And another challenge is also the non-compliance aspect. So anthrax is, of course, quite sensitive. If you've cut up meat and distribute it to your community and then people become ill, or you've distributed it into a water body and people become ill, or the environment and more livestock die, that's a sensitive issue. So, of course, people are reluctant to share the information. But remember what I said about the history being key to identifying and confirming these outbreaks and confirming them, so and managing them. So having strong relationships with the community are really important for ensuring that there is compliance and information sharing and openness around these outbreaks. And that also contributed to unwillingness to provide samples. So you would have seen that far fewer samples were collected than people who were likely to be infected, and that's just because people were refusing to provide samples. Again, the importance of strong community relationships. So we're going into the closing time, and I'm saving the best bit, I guess, for last, but also the smallest bit for last, because what is known about anthrax in Somalia, the answer is really there's not a lot published. Again, you would have personal experiences, but I wanted to share with you what's in the literature so that you can be inspired to continue to build this area of work. So a few years ago, we looked into the volume of zoonosis research in the Horn of Africa. And Somalia was one of the studies, one of the countries that we looked at. And the diseases that you can see here are all the diseases that were the top most studied in the region. And the spider diagram helps to understand the number of publications by disease. So you can see in the Somalia context, there's been quite a lot of research on brucellosis, maybe gastrointestinal bacteria, RVF. But when it comes to anthrax, there's really not a lot of research that's been done. And that's actually true across the Horn of Africa. It is a research gap where it's quite unusual. There's quite quite a discrepancy between anthrax being seen as a priority zoonosis, but actually data from the region being very, very limited, both in published studies as well as surveillance. So let's look at the 2 studies that were published in Somalia. So this is the first one and appears to have been published in a Czech journal. We found when we were doing a lot of this research, so much of the research is published by foreign researchers, not by Somali researchers. That's true of all of the zoonoses, not just anthrax. And this entire paper is written in Czech, apart from the summary, which has some important clues. It says the authors present a paper on the causes of a dramatic form of anthrax when the contagion was caused by the leather souvenirs from Somaliland. So that is confirmation that people had been exposed through Somaliland, but what's not clear from this paper is whether or not those people were diagnosed in Czechoslovakia or if they were diagnosed in Somalia. We've also found that a lot of the research on zoonoses in Somalia have been diagnosed in a different country, in this case, Czechoslovakia, by the looks of it. So that's all we know from that one paper. That's half of the research that's been published on anthrax in Somalia. The other half, quite interestingly, again, some researchers who are based in Kuwait, but possibly with a Somali connection. Perhaps these are people known to you. But again, quite dated, and this is in the Journal of Camel Practice and Research. And what's quite interesting about this is I've just included the snapshot. The whole paper was describing pastoralists and their knowledge in a variety of areas. It just singled out anthrax as being the disease of particular importance. They mentioned the local name, as you referred to before. I think you pronounced it as Kud, meaning southern death. And quite interestingly, it describes the fact that the pastoralists were managing this in a suitable way. So the carcass of any animal that died suddenly of suspected anthrax was covered on the spot with branches and then lighted up with fire. Nonetheless, though, they weren't aware of this environmental resistance. So they seemed to have some sort of practice that was consistent with knowing about the environmental connection, but perhaps didn't really understand what that was actually about. I'm pleased to report, and I don't know if Farah's online, he wasn't when I started, that we've also been able to fund some research on anthrax, not only in Somalia, but in the Horn of Africa through the Horn Project. And so Farah, who at the time was a lecturer with the Red Sea University, was able to lead the implementation of this project within the context of Somalia. And the aim was to assess the knowledge, attitudes, and practices of high-risk groups. What I'm presenting now is just from the farmer component, but there was some other data collection on abattoir workers and butchers. So Farah, as I said, undertook this study in 2 different areas of Somalia. Each of the different countries implemented it in 2 different areas. And the Somali population, so the questionnaires were undertaken with 381 people, most of whom were pastoralists and agro-pastoralists. So this was designed using random sampling and was designed to be a fairly robust estimate of the areas under study. The key findings were that anthrax was a disease that was commonly identified. So it was actually when participants were asked, do you know any diseases that are spread from animals to people? Anthrax was the most frequently mentioned by people, followed by tuberculosis, brucellosis, and rabies. They also mentioned the local name, Kud, and around a third of them said that they could recall particularly involving but similar to the study that I reported before, only 16% of participants linked the disease to the environment. So although they seem to recognize the disease potentially, they might not associate it with a disease that's linked to the environmental conditions. However, when digging a little bit more into their knowledge about anthrax, it became clear that there were quite serious knowledge gaps. So when participants were asked to name clinical signs, it was only around 40% of them that identified sudden death as being a common sign. I mean, that's not a bad number. But bleeding was only recognised in 14% of the, or named rather, by 14% of the people. And there are a range of other symptoms and signs that people identified as well. None of the participants recognised involvement by wildlife, which I guess might be consistent with the context in Somalia. I understand you probably don't have so many herbivorous animals ranging in rangelands there, unlike other areas. But where things got particularly problematic was thinking about what are the signs in humans. So you can see there that if they were identifying signs, then they were identifying gastrointestinal signs, but there wasn't necessarily recognition of cutaneous disease. And also quite interesting, I guess, is perhaps the stigma related to eating animals that have died. We suspect in Somalia that might be quite different in the context of Muslim populations versus in Christian populations in Ethiopia, for instance. So it might be quite uncommon for people to eat and distribute animals that have died in Somalia. And again, measures listed by participants that they use when handling anthrax or animals infected with anthrax. Not a lot of them really had any sense for how to manage these outbreaks. So the conclusion would appear to be that there's limited knowledge of anthrax among pastoralists, and I guess there's question about whether there's possible confusion with other livestock diseases given the descriptions of different clinical signs. But certainly the limited awareness or understanding of the disease in humans. So that speaks to the clear need for community awareness and improved multidisciplinary outbreak response. But definitely I'm keen to hear your perspectives on that and your interactions with pastoralists and whether or not these findings would agree with what you're experiencing in the field. So this brings me to my last slide, which is just to really reinforce some of the key messages. So anthrax is, of course, an important and serious disease that affects humans and animals, particularly herbivores, but not only herbivores. The key manifestation to be aware of is sudden death in animals with bleeding from orifices. If you observe that, you should know straight away not to conduct a post-mortem on those animals, and even a presumptive diagnosis would be better than a diagnosis that's confirmed but leads to environmental contamination. We spoke about the 3 forms of disease in humans, 3 to 4 forms, cutaneous, gastrointestinal, and inhalational, and all of those reflect the exposure route in humans. I've really emphasized this environmental cycle, noting how long spores can survive in the soil, and that's where environmental management is really key during the response. I've mentioned that it's one of the 7 prioritized zoonoses in Somalia, and of course that's why we're here. This is a series on the priority zoonoses. But I've also really highlighted the gaps in the literature that appear for Somalia in particular, and really encourage you to consider pursuing anthrax research as a topical area in your research agendas. Okay, thank you very much. to turn it back to you or stay online and for a facilitated conversation? I don't know.
Dr. Moore, can I ask you a question?
Sure, please go ahead.
Yeah, well, thank you very much for a really very excellent presentation. I've got two questions. One is, with this disease being so acute and animals dying suddenly, what is the target tissue? And apparently, I mean, the animal must sort of the toxin must eliminate the body's defense system in order to multiply and produce more toxins. So what is the target tissue of this toxin? And also, given that sort of you can see blood oozing from the orifices, I was wondering whether the cardiovascular system is the target. My other question is there any vaccine that can be used to eliminate the disease altogether, given that management of it may not be really easy, because the animals just don't develop symptoms long enough to be tracked and they die. Thank you very much.
Okay, do I respond to the question straight up? Is that
yes, go ahead, very good to, thank you for, this is the result of this insightful presentation. Immediately people are eager to discuss, and I like the question of the participant now. So go ahead to answer. Yeah.
So I am not a pathologist, I am an epidemiologist. So that first question is a little bit hard for me to answer. But my understanding, I mean, it's a systemic infection. I don't believe there's a very specific target, but I do believe that there is sort of a, part of it's sort of an immune-related, like a, what's it called? What's the cascade? Like it's the hemorrhagic aspect is related to the, what is the name of it?
The coagulation cascade. Is that the coagulation cascade?
Yeah. But it's not, that's not actually the name that I'm looking for. But it's the same as Ebola actually. But I can't quite remember what it is. But yeah, so I believe that the mechanism is stimulated through an immune-mediated reaction on the cardiovascular system, if that makes sense. And clotting cascade. So it impacts the clotting. So but that's not it. It's in COVID, sorry, I'm blanking. In COVID, it was also the same. This immune-mediated. If someone's listening and you're a pathologist, you can help me out. Otherwise, someone can quickly Google and you can type in these diseases and type in cascade and the word will come up. But yeah. Okay, and then the second question about the vaccination and elimination. So yeah, the animal vaccine could conceivably be used for elimination. But more often than not, the elimination is done in conjunction with environmental management. So the countries that have been successful in sort of eliminating have very intensely managed the environment, and the vaccine has been done sort of secondarily. But again, when we say countries that have eliminated, countries in the Arctic thought this was not a problem anymore, and now with melting permafrost it is. So, so I don't think vaccines would ever be used alone, and I believe they don't have a long-term immune response either. So in Australia, I believe they're used annually. So it's quite a resource-intensive process, and more often than not, to eliminate anthrax, it would be done through, creating enclosures to stop animals from grazing in there. That makes sense. But in combination with vaccines in areas around there and in seasons that are typically regarded as high risk. I hope that answered the second question at least. Has anyone Googled?
Yeah, question.
Someone by the end.
Doctor, do you hear me?
Yes, now we have who is Gullet? Gullet is have hand. We have you can unmute.
Thank you very much for your wonderful presentation. My name is Guled Abdi Ahmed. I work at the Ministry of Livestock in the state of Somalia. So my question simply is, as we know, some of the infectious diseases has, or have a rapid confirmatory test that you can use in the field on, So my question is, does this particular anthrax disease have that kind of confirmatory rapid test that we can use, the field by the, cows or veterinarians? Thank you.
Sorry, can you repeat the question? I only caught the end around veterinarians and cows. So what is the question related to those, though?
The question is that this, anthrax disease has a rapid confirmatory test.
Rapid coordination? Is that the word that you're saying?
He's talking about
Confirmatory test.
rapid diagnostic test as issue of diagnosis. Rapid diagnostic confirmatory test of anthrax.
Rapid diagnostic test. Yes,
Yeah.
yes. So you're asking, can vets and cows use the diagnostic test? Is that what you said?
That is actually available,
Yeah. So I can't answer that in Somalia. I would suspect not. And the rapid diagnostic test has not been widely used in African contexts. In more circumstances, CDC has actually been supporting labs to develop PCR capacity. And I'm not sure if that's because the rapid test is not seen as I know that in Australia it has some use as well, but it's sort of, they go quite quickly to other methods of confirmation. So in some ways, the value of a rapid test, when you can't confirm it, I think is a bit tricky, given that the signs themselves are also quite, should lead you to be suspicious about anthrax, right? If it's a sudden death and there's bleeding from the orifices, a rapid test in that context is probably not so helpful. What you need is a confirmatory test, if that makes sense. But does anyone on the call know if they're available in Somalia? I don't believe they're available in Ethiopia, the rapid test.
Okay. So thank you, Gullet. Can we go to Abdurrahman Berry? Abdurrahman Berry, you can unmute and ask.
Thank you very much. Thank you very much. I am Abdurrahman Dahir Barre, Masters in Veterinary Pathology from University of Nairobi, Kenya. Thank you for your presentation.
Very welcome.
You need to answer the last question.
Even I can answer the question from the other guy. The guy was asking for which system that can affect the disease. I can answer the question, huh?
You are
And
welcome.
also, he was asking for rapid test. In anthrax, we cannot use for rapid test. Only we can use for confirmatory diagnosis that we are using for BCR or biochemistry. Only that. That is for the answer of the other guy, Goulet. I don't know I don't remember his name, huh?
That's because it's not available, right?
Yeah, it's not available in Somalia.
Yeah.
Okay, thank you very much. I'm also working for Anthrax now. I'm a lecturer at University of Horset. We are working for research concern for Anthrax this year in Johar and also some Shabele. So thank you very much. I'm having one question and comment. Let me start for the comment. I'm telling for all students who are studying for veterinaries, there's a little bit of gaps of anthrax. So I'm encouraging for all students in Somalia who are working or who are doing for bachelors, please, you have to conform or you have to do anthrax. It's a lot of gap. As you have been seeing the lecture of these guys, they told us there is no research in anthrax in Somalia. So I'm encouraging for all students in Somalia to focus the anthrax, and it's a zoonotic disease that can be affected even the human being. Thank you very much. My question is, how can we prevent or preserve in biosecurity when someone is working in lab, anthrax, and also in working in field? I think you understand my question.
Yeah, so it's typical barrier control, and so gloves and a suit if you're able to wear a hazard suit, as well as then respiratory protection, which would cover the mouth. And a face shield would be ideal as well, because you can get infected through splashes and mucosal contact as well. So that is really just informed by knowledge of the transmission pathways. And in the laboratory context, it's I believe it's a BSL-3 pathogen. You would know more again because you're a pathologist. Generally it's not recommended that people handle cultures. It's considered a high risk laboratory exposure. So it's often people, if they're handling samples, they need to take precautions because they've somehow been informed that they're receiving samples that might be anthrax, So again, as veterinary practitioners, or as someone who might be submitting samples, if you're writing on the outside of the sample, suspect anthrax, and double bagging and taking all those sorts of precautions, and that's ideal.
Thank you very much.
Again, I fully acknowledge that in the context here, it's not going to be everyone that's collecting samples and submitting them to the laboratory. And in the absence of laboratory capacity, even at a federal level, collecting samples might not be a priority. So I think the key message would be, don't collect samples if you can't do it safely. And it's better to have a suspect diagnosis than a confirmed diagnosis where you've exposed the environment and people and other animals in order to get that confirmed diagnosis.
Thank you very much. What about in Australia? How do you use in Australia?
How do we diagnose?
Yeah.
It is through the methods that I've said. So confirmatory tests using PCR and culture, but there the system is sort of dependent on your average vet who might have come out and been suspicious, calling the government vet. So the private vet who would call a government vet, and that government vet has more specific training on managing and handling that kind of herd level outbreak. And it's the government vet that would take the samples and enact movement controls and all of those things. So the average private veterinarian in Australia would not have capacity to manage an anthrax diagnosis or even an outbreak response. They would
Thank
do
you
that
very much. Thank you very much. I hope you will share us for the slides. Thank you.
Okay, Dr. Abdurrahman, can you please, do you want to ask something about the pathogenesis? The first question, about the toxicity, once you are a pathologist? Do you want to add something or?
Yeah, thank you very much. I don't want to ask you more questions for our lecturer. So
no, I mean the first question of asked about the pathogenesis of the toxicity happen for the hemorrhages. If you have something about pathology of this issue to say. Okay, thank you. Thanks. So let us go to Abu Bakr Ahmed. Abu Bakr, you can unmute and ask your question. Abu Bakr, are you around?
Okay, thank you, Professor.
Abu Bakr? Yes, your voice, please. I'm not listening. I hope Dr. Siobhan is listening.
Thank you.
Okay, Abu Bakr.
It's a good presentation. My name is Ahmed Abu Bakr Yusuf. I am a student at Nairobi University, Master of Veterinary Public Health. My question is, as we know, this disease is the dangerous disease in human. My question is, which way or the best way to prevent this disease, the people of working laboratory and the people of working slaughterhouse? As we know, the people of risk this disease. Can you tell me the way or the easy way to prevention this disease? Is my question.
Thank you. You can hear the answer.
So
Let's go
the
ahead.
prevention in the laboratory setting is the same as I just mentioned before. So cutaneous barrier, respiratory protection, and ideally a sort of suit if possible, biohazard suit. In the abattoir setting, it is a lot more challenging, but you still approach it from first principles, right? And it wouldn't only be anthrax that you'd be worried about in that kind of context. There'd be brucellosis, which would be much more likely to be aerosolized and cause abattoir-related outbreaks, right? So it's a lot harder in abattoirs, and I think most countries have generally tried to tackle the aspect of prevention at the farm level, trying to stop animals that are diseased getting slaughtered and going for slaughter in the first place, which again I suspect would be quite challenging in Somali context. So I think again, if you're thinking about research directions for zoonoses in general, doing surveillance and working with abattoir workers and looking at the types of exposures and diseases that they have in that context, I think would be a really rewarding way to get quick information from high-risk populations quite easily in Somalia. Doesn't really answer your question, but the point is it's quite hard in that context, and so the prevention is really at farm level.
Thank you for your answers. Just satisfied for your answers. Thank you.
Thank you so much. So let us go to Ziad Abdullah. You can unmute and ask your question, please. Ziad, are you around? Ziad Abdullah. So before, okay, welcome. Welcome.
Thank you, Master Moderator. Thank you, Dr. Siobhan. I really appreciate you, how you are, and update us in anthrax diseases. And my name is Ziad Abdullah. I'm a lecturer in Jijiga University and in Ethiopia, eastern of Ethiopia. I think most of the, Somali community, whatever they are, whether it is Kenya or Ethiopia, in Somalia or in Somalia, they are the same environmental. The people are pastoralists, they moving one place to another places. So if if one of the pastoral community have a problem, they also share that problem with others. So when I come to my questions, I need particularly the key message for your presentation, the key message that are concerned on this disease prevention during climate change. For example, during drought seasons, most of the animals they become immuneless because their immunity are decreasing. So at this time, most of time this anthrax outbreaks at that time it will start. And the second time also when there is heavy rainy seasons, because the bacteria which are causing the anthrax are spore forming bacteria. So and it can this bacteria can live in the soils many years due to their spore forms. So at that time when there is a heavy rains, heavy rains will there is a lot of outbreak at that time. So also the pastoralists have that problem in that seasons, during the dry seasons as well as heavy. So that prevent this seasons. Another thing is, in the collaboration of many institutions, in the collaboration of Minister of Life, Minister of Health, as well as the environmental.
Yeah, the line, I think, I hope you will listen to some of the question because the line, I think, is cutting from my side. I hope you will listen some. Mr. Ziad, are you still around or Mr. Ziad? Maybe we lost him, I think. Okay, so before Ziad come back to listen to his answer, can we have another question from
So
because Ziad,
I
Ziad,
back
are you
sorry.
around? Yes, please. I
Allah
hope
Ta'ala
even
Farad, you-
yes. So
Yeah,
bring
yeah,
the yeah, yeah.
yeah.
Okay.
And you know, and another thing, another my question is, what is the important message towards different institutions who are, you know, who are the mandate is to prevent the diseases, such kind of, not all anthrax, but most of the zoonotic diseases. So, for example, the Minister of Health, the Minister of Livestock, and as well as the Minister of Environmental. So, what are the key messages towards their collaboration? Because they didn't collaborate, most of them, all of them, they do have different mandate, and so they do not join on the on the border of the mandate. Most of them, what is the meaning? How much? Because your presentation must be contribute to something to the Somalia.
Yeah.
So, and we need only that message, as well as, and the last question, and in pastoralist we have, problem of movement, one country to another country, one place to another country. We can prevent some of that. This is, most of the, this is certainly killing of the livestock. So, there must be, in order to prevent it, ring vaccination, not annual vaccination. So we do not have a calendar vaccination. So most of them, whether it is Somalia or Ethiopia, they don't have in calendar vaccination in a pastoral community. So what are the suggestions toward this? For example, annual vaccination or calendar vaccination in order to prevent it, the disease. Thank you very much. I finished my question. Thank you very much.
Yes. So, Doctor, we have 3 questions from Dr. Ziad. If you can, I think the middle one, how the institutionalization, I think you can start with then the climate change effect and the general issue about prevention about all zoonotic diseases, including
the second question.
Yes, about ministries and institutions. How yes, that's very, 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 very interesting. So it's clear
the third question.
Yeah.
Yeah.
Yeah, she's going to start to answer. She get your questions now, Doctor.
Remind me what the question was on climate, though. I only heard.
Okay. Yes. The first question he asked about climate change. He wants keywords, keywords for climate change of anthrax issues. Then he come back and asked about institutions, particularly for so it is important, he said, for Somalia, and then he asked about the general prevention method for anthrax and other zoonotic diseases. I think. Am I right? It's okay like that? Okay, go ahead.
Yeah, no, I missed the climate one because there's not. So I heard it more as you were describing the conditions in which it happens, and as you've said, and as I said earlier, it's associated with both wet and dry, and it depends on the different areas. It's clearly connected to climate, although the pathways are just complicated because so much of that relates to human behavior as well, which move livestock around and which lead to consumption of livestock. So it's not as black and white as just saying it's related to climate. But with respect to climate change, there's not research that I'm aware of that has made predictions on this region in terms of how it might be impacted by climate and climate change, and therefore whether or not we would expect to see it be worse or potentially better under climate change scenarios. The research that I did present is largely focused on the Northern Hemisphere, and also particularly the Arctic area. So again, it's a research gap that people on the call might wish to pursue, but realize that one of the limitations is a lot of those studies rely on decades worth of data, and they can be identified spatially and then identified in time in relation to climate events. And so in the absence of strong surveillance systems, it's quite unusual in Africa to have decades worth of data through which to do these types of studies that rely on good disease data. The climate aspects is very easy. It's the disease outcome data that's a bit harder. With regard to key messages around coordination, so as I understand, there's now the high-level One Health technical working group that's been re-established at the Somalia level nationally. I don't know exactly that they have figured out what the role will be with respect to coordinating outbreak response if that were mounted in Somalia. But in the context of launching a response and coordinating for response, I think anthrax over every other disease is quite easy to justify the need for human, animal, and environment sectors to be present. And so, as a way to start fueling the response, the cooperation that happens across sectors, I think anthrax is should be a priority for establishing those coordination mechanisms. The reason why I say that is I see in Ethiopia, rabies get talked about a lot as being, on the One Health Coordination Committee. But at the end of the day, the people that are going out and delivering that intervention are veterinarians doing animal vaccinations. So, of course, it's very clear that rabies is important from the human health point of view, but the people that are doing something and out, implementing a response are on the animal health side. And that's quite different to anthrax, where there is very clearly a need for humans to go out, human health professionals to go out and is that directed at me?
No, go ahead.
So human health professionals need to be there to investigate and enumerate the number of people who have been affected. Animal health professionals need to go out there to investigate the number of animals that have been infected. People who are involved in rangeland management, environmental management need to go there to be involved in the response. So I think anthrax is a really great way, actually, to and starting point for establishing the first strategic plan for disease management and control in Somalia. Although I know that you actually have drafts of several of these, and you might not be implementing those strongly.
Okay.
There's a third.
Okay, thank you very much. Thank you very much, Doctor. Yeah, but in Ethiopia, the people are living in Ethiopia, they do have different agroecological. Some of them are pastoralists, their livelihood are dependent on livestock. Some of them, they depend on, agriculture. Most of the pastoralists, but it depends on only, livestock. So, we do have a problem in, annual vaccinating, because the pastoralists may.
Sorry, go ahead, Ziad. Unmute yourself, please. Ziad? Yes, go ahead. Go ahead. Sorry.
The pastoralists do not have a permanent position.
Okay.
Annual vaccination is not applicable. So, they have a problem. So, if there is any forward, that can you, that
I'm not
Anthrax.
breaking up quite a lot.
That
Yes.
You mentioned that at that point.
Okay, thanks. So let us, I don't know, just your question is very good, but the line is cutting always and the words is not clear. So let us hear from Muhammad Abdi, then we will come back, inshallah. To Muhammad Abdi, if you are around, please.
Oh, yes, Mr. Coordinator. I'm sorry, I was the first person who asked you the question without
yes,
seeking your permission. My
Yeah.
apologies.
No, you are welcome. Your question was very
Yeah.
interesting, and it's
I'm
okay.
still not
and
sure that you have the answer.
And you are very keen, yes.
Yeah, no, that was
Yeah.
that was a really great answer, and I realized that as a pathologist, I assumed that Dr. Moore was a pathologist, but apparently, because I came late to the meeting, I didn't realize you were epidemiologist. My apology for that.
No.
But the reason I came back to it is really, I am a veterinary pathologist, although I have left the field of animal diagnostic pathology a long time ago, and I only work on toxicological pathology in the drug development. I was really fascinated by all the answers that were being given in terms of the pathogenesis, and I realized this is really a very, very rich area of research in terms of pathogenesis, because I will Google it when I leave the meeting, but I think that this disease does cause either coagulation problems, destroys the blood vessels, so blood goes out, knocks out the immune system at the first instance, so that the animal cannot respond to the anthrax, and then the bacteria has a lot of opportunity to multiply without hindrance, having knocked out the neutrophils, lymphocytes, macrophages, all of these things. So I think it will be a very interesting area of research for those of you who are involved in animal veterinary diseases. That's all I came back to say, and again to thank Dr. Moore for really excellent presentation.
Okay, we have Abu Bakr Siraj also. Abu Bakr Siraj, please briefly your question or your comment.
Thank you very much. Thank you, Siobhan, for the wonderful presentation. My name is Abu Bakr Siraj. I am from northern Kenya. I think this presentation was very informative and educative. I wanted to contribute to the question that was asked by the participant, because coming from Kenya, sharing the border with Somalia, sometimes we also have this issue of because of the porous border, the animal moving from Kenya to Somalia. Before I ask a question, I just wanted to also contribute to the question that was asked by I think Mahmoud about the pathogenesis of this anthrax. I'm a veterinarian. I also did masters in veterinary public health from University of Nairobi. I think that one of the anthrax spores or toxin is very powerful. The powerful toxins that actually causes what we call a hemodynamic factor that actually causes the vasodilation or the cardiac dilation. And because of that, they reduce heart function. And then from there, they also cause damages to the blood vessels or blood cells, which actually lead to the splenomegaly and pooling of blood from all the body openings. That's from the pathogenesis. In terms of the rapid test, so I'm just trying to compare the Kenyan context. In Kenya, we don't have the rapid test, but you can collect or you prepare a blood smear. You prick the ear vein from a fresh carcass, because actually the carcass, because of the rapid decomposition, they decompose very easily or very quickly. You prick the ear vein, you collect the blood smear, you air dry, you do the GM staining. From there, then you can do your microscopic examination, and you can able to actually confirm whether you can see the Bacillus anthracis from the laboratory test or the microscopic test.
And that's, I just ask, intervene? Is that done at the regional level? Yeah.
Yes, we do have the regional level, regional veterinary laboratory here, even in Garissa. Garissa is just a few kilometers from Somalia.
Mm-hmm.
And we also have at the national level, the Central Veterinary Laboratory. They can also do that test.
Sorry, go ahead.
Thank you. My question, we usually have this mass camel death.
On that side, may I have a question?
Just wait for Abu Bakr Sarraj to finish, please.
Thank you.
Please, okay, thanks.
Thank you. We also have an issue of mass camel death along Kenya and Somalia border. We had, I think, in November, December, there was a mass camel death in Doble, and then because of issue of the nomadic life and our porous, our border is just porous. It's just an imaginary line. So these persons move in and out, especially in search of pasture and water. So we had mass camel death reported in Doble, and then they cross over to Kenya, especially along the Liboi part, Wajir South part, Ifif. And from the pastoralist perspective, they usually call it in the local name Kut, which is actually the anthrax. But I don't know whether the Somalia team have collected the sample from their site just to confirm whether it is actually the anthrax that was causing the mass deaths in camel. When they cross over, we try to, the team from the veterinary team from Kenya, they try to collect sample. But unfortunately, they did not get carcasses to confirm actually it was an anthrax. But they got a sick, few sick animal. They collected the serum and the whole blood, and also they made a smear, which actually from the lab finding, I found that it was the tick-borne diseases were called anaplasmosis and also the thrips, which is also another hemoparasite that are transmitted by sesefly. But from the farmers' perspective, when they were reporting along the border, they actually saying that this looks like an anthrax because of the sudden death, part of the sudden death. And even though we didn't confirm that there's actually that oozing of blood from the body opening, that blood discharge. In terms of also anthrax human cases, you know, our culture, Somali culture, is that once the animal is dead, we don't usually consume the animal from the meat or anything, animal product from the dead animal. That is why we also thought that we rarely get sometimes the anthrax human case, especially in our pastoralist. Even though most of them, they are also well conversant with the anthrax, especially the continuous form, which is a most common form as you put it across, and also most common form also in Kenya. Some part also, we also get the GIT form, especially in the central part of Kenya, which is especially Muranga, if you are a vessel with around some area around the Nairobi. Because these guys, whenever they see any carcasses, they usually celebrate because they see that's a good meat or a good mboga, they call it there, just to feast on. And that's why my contribution from my side. So I just wanted an insight, just a bit of contribution for my colleagues from the other side, because we share this culture, we share the geographical location in terms of the coverage. I just wanted to, I know in Somalia there is a lot of gap in terms of the zoonotic diseases, the research and everything. There is a gap, I know. Kenya, the anthrax is top, the number one in terms of the zoonotic diseases. I think Kenya, we prioritize 36. Anthrax is top, number one in terms of the ranking. So that's why currently, in terms of I'm supporting the community-based surveillance for zoonotic diseases along the Kenya-Somalia border, along Kenya and Ethiopia, and also in terms of cross-border counties. We also we also have a Turkana County which borders South Sudan. So Ethiopia, because I know you're working in Ethiopia, we'll also have some cross-border coordination meeting with Kenya, because I am in Ethiopia, because GHSA is actually implementing the community-based surveillance in Ethiopia, Kenya, and other countries. But Somalia, because currently they have actually prioritized their zoonotic diseases, I'm sure they have. It's a long journey, but we will have also Somalian plan. But currently, I just wanted to have to hear from the other side what is happening along their border, the Kenya and Somalia. I don't have question. It's only my contribution. Thank you very much.
Thank you. Thank you so much.
I can also reflect, though, that what you described with the camel outbreak was very similar here in Ethiopia. We had the same mass camel death, and still the pastoralists themselves were saying, we're associating it with anthrax. Although when you listen to the signs and symptoms, they weren't characteristic of anthrax, and it never was confirmed as anthrax, and never was confirmed either way. But other possibilities were obviously pastoralosis and that other disease, top priority disease, RVF, as a possibility as well, but never got a diagnosis. But the conversations were very similar to what you mentioned there for Kenya.
Okay, thank you, Doctor. So before we go to someone from Somalia, if you want to comment to answer for Dr. Abubakar Siraj, briefly your question, please, Mahmoud Abdi, just briefly, because we have many. The presentation is very rich, so the discussion also is very rich. So maybe we need to have another day for anthrax, another from our neighboring country, our Nairobi border people, like that. So Mahmoud Abdi, please briefly your question, just directly go to the question or your comment, because we need someone to talk to Abubakar Siraj, someone from Somalia, if he has any information about the outbreak in the border there. Mahmoud, go ahead.
Sorry, I don't have any question. Maybe I didn't remove my hand from the screen.
Okay, someone asked about, he has a question. Did we lose him? I'm sorry.
Yeah, no,
okay.
I'm going to remove my hand.
No problem.
Yeah, thank you very much.
Yeah.
Thank you very much.
I have a question about Abubakar. Abubakar, he told us the border of Kenya.
Sorry, we have two Abubakar. Abubakar Siraj, you want to reply for Abubakar
Siraj?
Guy
from
right?
Kenya.
Okay, go ahead.
Yeah, thank you very much. So he was saying there's a diagnostic investigation in the lab of Kenya, and they were doing for some lab tests from the screen test, and they are doing for the blood from the geography, the fresh carcass to the Jimsa stain. So I need to be asked for question for Abubakar. Is that a confirmatory diagnosis or a final diagnosis? I need to know that. As a pathologist, we don't know how to make an screen test to that side of the anthrax in animals, whether it's a human or animals. Thank you very much.
Okay, so Abubakar, please briefly go answer him, because we need someone to comment you. I know your main question is what is going on in Somalia side about anthrax. So
I can elaborate a little bit on that, actually.
Okay.
He says, what is considered a confirmatory test? And then, so culture is obviously the gold standard. And when we were publishing the findings from the PCR, we were challenged on whether or not that was a sufficient confirmatory test. But most of the literature, and even WHO and WOA these days, do suggest that PCR is sufficient for confirmatory diagnosis. So it's not considered screening. It is considered. It has been in the past considered only sufficient for screening, but increasingly literature is suggesting it is confirmatory. Again, that requires a lab with significant capacity, but a lot of PCR machines moved up into Somalia through COVID, so I would expect most countries now would be in position to roll out PCR diagnostics, unlike it might have been 3, 4, 5 years ago. So maybe not at the animal health side, but
okay. Okay.
Thank you. Let me just respond. You know, there is a difference. There is a difference between the screening test and the laboratory test, which is actually the confirmation test. Because I said we don't have the rapid test. Rapid test is a screening test, which is the screening one. For Kenya, there is a culture method. You can do the culture method. You can do the serological test and the molecular technique, which is the PCR. So the one that I said is that you can collect a blood smear from the ear vein from a carcass that you suspected that is actually that died from the anthrax. From there, now you can take to the lab, dry elect to the lab, and do the actual confirmation from that blood smear. So that's not a screening test. It is actually the laboratory, the confirmatory test.
Thank you. Okay. So anyone please about Somalia border, about the outbreak of deaths of camel. Anyone want to respond for that? Any checking or any investigation took place
I
there?
hope you are hearing me very well.
Okay, thank you. Welcome, Dr. Ahmed. Yes, very good to have you. Thank you. Welcome. Very good.
Yes. Thank you very much, Professor Siobhan for the nice presentation, and thank you for the participants for the good questions. Regarding the Anthrax in Somalia, already talked by Professor Siobhan. It is not put on the light, even as we did many researches on animal and human, we did not focus on this side. We must probably focus on other zoonotic pathogens, most probably on vector and vector-borne diseases. But as you have encouraged it, we will look again for such diseases, as we already started it with these webinars, as it's already prioritized in this year in Nairobi. Regarding the camel diseases or camel deaths, before the camel deaths and for the anthrax, yes, we don't have much researches or much data, but as in the CAP, you have seen that the people, they now most probably the disease. So the disease is there, but needs more attention to focus on it and look of it, because we heard about while I was working in the ICRC, I have heard about some cases about anthrax, but did not confirm it. Regarding the camel disease, yes, I heard some camel deaths from the border, and even in the southern parts in Bedawa and others. It is not confirmed, but it is most probably suspected as a hemorrhagic septicemia from our side, but it's not confirmed. Thank you very much.
Thank you, Dr. Ahmed. And also, just I want to add, we have 2 students of graduation thesis from Abrar University. I'm supervising them. They collected samples of camel from Kismayo. I don't know what it is. Maybe Kismayo is more further the area. Maybe we are talking about. We checked just like Dr. Abu Bakr Siraj said. We checked the blood films, and really we find pyroplasmosis and trypanosomosis and anaplasmosis. And of course, already I learned from the lecture of the presentation of Dr. Siobhan that already, of course, sudden death is the cardinal signs of. Therefore, once you sample living animal, you can suspect other diseases than more than to suspect anthrax. So once we are sampling living animals, of course, we are going just to find this disease, but not anthrax. Anthrax is much of just after this clinical science presented here. It needs confirmation because clinical science is very significant, I think. Thank you. So I agree with Abu Bakr. We have blood films, but it is pyroplasmosis, trypanosomosis, and anaplasmosis. Thank you. But I respect the point of hemorrhagic septicemia for the if even there is sudden death there. But yes, our problem is that. But thank you we are going to concentrate, try to sending students, researchers to try to do something, as Dr. Ahmed said. We want to go further for this disease like anthrax and other diseases like this already neglected in Somalia. Thank you. This is very productive discussion, and of course, is there anyone want to, because we don't want to lose any. Sorry for your time, Dr. Siobhan, but the discussion, I like it. It's very productive, really. Anyone want to ask him before we give Dr. Siobhan the last chance before closing? Okay, so do you want Dr. Siobhan say, okay, you can go for the another last key messages for 2 minutes before we close.
Just 1 minute, Dr. Abdullah.
Okay, you are welcome. Thank you, Dr. Abubakar.
My network was,
it is 1 minute. It is 1 minute, as you said. Okay, not me. You said
yeah, 1 minute.
Okay. Okay, go ahead.
My network is a bit intermittent. You know, interesting part is that we actually, part of the intervention, the team vaccinated the camels against the anthrax and black water. And the response was positive. It was, in fact, most of the camel that were vaccinated, from their findings, that were vaccinated against anthrax and the black water, because we have a combined vaccine, the anthrax, which is actually effective against anthrax and black water. So from the report, the findings from the field, that all the camels that were vaccinated against the anthrax and black water were responding very well. In fact, from the area that were affected, that was they did an emergency, the firefighting emergency kind of vaccination. So that's why I also wanted to hear from the other side. And the good part is that the Somalia part also, they are sourcing the same vaccine from Kenya. That's what I had. So I wanted to also share this information in terms of One Health along the cross border. The communication part is very important, so that we can also, whenever there is such kind of disease affecting livestock, even the human, we can share before they spill over to human. Thank you a lot for your time. I am looking forward to have another, I think, wonderful and interactive session with the One Health Somalia, One Health Field. Thank you very much. Thank you,
Thank
Prof.
you. Thank you. And we will communicate you personally for this to cooperate on this issue in the Somali-Kenya border for anthrax. You will be very helpful for us. I am sure of that. Thank you. Thank you. We have your email already, I think, here in the list. So we will communicate, inshallah. Okay, so Dr. Siobhan, is it 1 minute or 2 minutes before we close?
No, I don't. I think I've said my last word.
Very good.
A lot of expertise in the room, so in the virtual room anyway. But it's really great to hear about the cross-border interest and cross-border collaboration. That's obviously so critical.
Yeah.
So thank you very much for your engagement. Well,
thank you so much. And with your collaboration and Dr. Abubakar, we will try to do something in that area, inshallah. With your help, it will be okay. Everything will be okay. So again and again, thanks a lot to Dr. Siobhan, and thanks for all the participants. Thank you so much for being here, and I wish we will see you again. Of course, we are going to plan to see again, if not in anthrax, in another prioritized disease in Somalia for the year 2023. Have a good evening, have a good day, have a good night, everybody. Thank you. Thank you. Bye-bye. Bye-bye. Bye. Thank you. Thanks so much. Bye-bye.